So that's a great introduction into our clicker questions for today, which are, number one, telomerase is a DNA-dependent RNA polymerase, DNA-dependent DNA polymerase, RNA-dependent RNA polymerase, RNA-dependent DNA polymerase, or B and D.
What do you mean by dependent here?
Could we reason that it's DNA dependent too?
10, 5,
Oh my God!
this is why I count down for you. So I heard some discussions, poor people in the front row actually will listen to what you're saying, and then I'll repeat some of it, but I won't mention who's saying what. The big question here, actually the two kind of questions, what's a DNA-dependent or an RNA-dependent polymerase? And then, of course, what is the polymerase? So we'll just start with the polymerase part here. Anything which is an RNA polymerase does what? It makes RNA, and the same thing for DNA polymerases, it makes DNA. Okay, so what do telomerases do? They make what?
They synthesize RNA or DNA.
Okay, do they synthesize RNA or do they synthesize DNA? Okay, what is a telomere? It's the ends of the DNA, and so it's extending a DNA. So it's clearly a DNA polymerase. Okay, so that eliminates A and C already. Okay, so B or D. Now what about this dependent? What does that dependent mean? It means I won't repeat that. So what it means is, what is it using as a template? So what's it using as a template here? It's RNA, which it's using as a template. Is it using DNA as a template? Does it use DNA as a template? No, it doesn't use DNA template. And that's actually really strange, because almost all reverse transcriptases do use DNA as a template as well. And we'll see that towards the ends of today. And of course, all of these wonderful enzymes are present in viral replication. So you have lots of viral replication as well. Yeah? Okay, so the question up here was that they thought that there was actually using some of the DNA template to make the copies. No, it's just the RNA template, which is part of the telomerase. So another kind of question you could ask here is, what's the telomerase made of? And the answer is protein and RNA, because the RNA template, which is used, is part of the telomerase enzyme. So it's both the RNA and the protein, which is important for that. Okay, our next question for today, moving on. DNA damaged by a deamination of guanine is likely to be repaired by proofreading, base excision repair, nucleotide excision repair, non-homologous end joining, or mismatch repair. And by the way, we did not talk about deamination of guanine in terms of how it got repaired. This is what you're supposed to be thinking about, what these repair mechanisms do. $15, $10, $5, okay, so deamination of guanine. What does that give you? That might be in your notes. I forget if it's hypoxanthine or another unnatural base. But it's got a very similar shape and size to what you had beforehand, right? So is that going to cause a big change in the structure of your DNA? No. What does nucleotide excision repair usually recognize? Structural problems in the DNA. What about mismatch repair? Also, it's a structural change. It's recognizing that there's not an appropriate base pair, which is formed there. Okay, then we've got non-homologous end joining. What's that? Nobody liked that one. Anyway, it's for double-stranded break repair. So we're down to base excision repair and proofreading. Proofreading is doing what? It's a replication thing. When do you have proofreading that takes place? It's an incorrect base pair incorporation. So what does that leave us with? Base excision repair. So, and that's true not only because there's not much of a change in terms of structure when you get deamination, but also deamination is one of these things that happens spontaneously, and it happens really quite frequently. And so it's not surprising that you have base excision repair. But what does that mean about something that you have in your genome? What kind of enzyme are you likely to have in your genome if this does get repaired by base excision repair? You have a DNA glycosylase, which is exactly right, but what kind of DNA glycosylase? A DNA glycosylase that recognizes deaminated guanine specifically, like your uracil DNA glycosylase. So there are these particular enzymes which will do that and take care of it. Okay, so that's it for clicker questions. RecA und omnes, and we'll move on. So today we're going to talk about double-stranded breaks and transposons, the jumping genes. This is, in fact, exactly the same slide we had last time. Non-homologous end joining. It's one way of dealing with these double-stranded breaks. What happens is you have, again, a double-stranded break. Ends get chewed in by the Ku proteins, et cetera. So the way that you get antibody recombination that takes place, interesting gene modification. And you just pop those two ends back together. Big problem that you have here is you've clearly lost some genetic information when that happens. If you've got junk DNA, put the ends together, maybe it's not so much of a problem. But as we're finding, there's actually a lot less junk DNA in all of our genomes than anybody thought was there. So the backup mechanism, and a much more high-fidelity backup mechanism, is so-called homologous recombination, and that's where you use a similar piece of DNA, not always identical piece of DNA, to repair what you had back here. You have a question? No. Okay. So that process, again, is basically taking the copy of genetic information from the sister strand and using that to fill things in. At first, people only thought this happened in diploid organisms. Turns out it happens in haploid organisms all the time. why and how does that happen? How do you end up with homologous chromosomes? You replicate. And so whenever you're replicating, you're always going to have two copies right, some people will say, behind the replication fork, after where you've got your DNA polymerase, in fact, two different DNA polymerases, your leading strand, your lagging strand, all the other goodies that you have at the replication fork. As this replication fork moves along, in many cases, it's going to come across a nick in the DNA or some kind of problem in the DNA. If you think about it, your leading strand is going along here. Here's that template. There's a nick, which is just a break in that backbone. Uh-oh, we've got a double-stranded break here. And so this double-stranded break can get repaired by, fortunately, that newly copied lagging strand. And so you end up with degradation here on these broken DNAs. You always end up with this 3' overhang, and we'll see this as we come back again and again and again. So you've got a 3' end single-stranded DNA. That 3' ended single-stranded DNA can now go in base pair with the strand that it was homologous to in the first place. Now, that provides a 3' OH, and that 3' OH can extend, just like always with our DNA polymerases. Then you've got your lagging strand, which is getting longer and longer in this case, but you've got lots of single-stranded binding protein around. So it just basically waits until the primer catches up, and you have this restart of replication. And that's probably what most of this homologous recombination does. Most of the time, it's repairing these, what are they called, stalled or broken replication forks. The process that you go into to degrade these ends, et cetera, is a really interesting one, and at least in E. coli, Dr. Corsell in our department is working on it. Yeah, you had a question. This is for homologous recombination only. The non-homologous end joining is just chop in at the ends, put them back together. Here you leave a 3' extension on one of them. That then goes and pairs with your homologous, which is why it's homologous recombination, homologous strand on the other side. Yeah? So the question is, in fact, this happens in haploid cells as well as in diploid cells. We'll talk about diploid cells and meiotic recombination in a couple of minutes, but these are all in haploid cells as they're undergoing replication. Yeah, they're haploid standard genome, but they're always undergoing replication. And so in that case, you have two copies of one genome. Yeah, and that's clearly homologous. It should be identical except for the changes which happen due to your not incorrect replication and fidelity, et cetera. But that's, again, it's 1 in a billion that happens. It's pretty rare. Yeah?
We may be talking about this, but is there something that's keeping that 3' end from base pairing with itself?
I'm not. So the question is there something that's keeping that 3' end from base pairing with itself? You mean this 3' end right here? Yeah. Yeah. So the only thing that this could base pair with is this strand over here, because whatever else was there that it was base pairing to is gone. So unless it's self-complementary, which is then wrapping back, would be folding back on itself. And the way that stopped is, thank you for leading into the next slide, are these very specific single-stranded binding proteins, which are not the SSB that you have at replication forks. These are specific proteins that are involved in homologous recombination. And so that's basically what's stopping them. It's the binding of this single-stranded DNA binding protein. In the case of E. coli, it's called RecA. And again, all these things are named after the mutations. Rec for recombination. It's the first protein that they found in that process. RecA binds to single-stranded DNA in a very cooperative manner, a lot like what you see with single-stranded binding protein. But the really cool thing about RecA-bound DNA is that the base pairs are actually now available to interact with other homologous base pairs. And so that's basically what this double-stranded piece here, also called heteroduplex DNA. And heteroduplex is just one strand has come from one place, another strand has come from another place. It's not your normal homoduplex DNA, which is the double strands that you have. So it's a single strand which comes and binds, and is basically looking to find a homologous sequence to basically say, again, totally over-anthropomorphizing here, that this is the right place to recombine. This is a homologous DNA sequence. So RecA bound to single-stranded DNA. Once it finds an appropriate homologous sequence, then you have this ATP-dependent process. We have disassembly, and now what happens here is what's called strand exchange. And so you started out with a single-stranded piece of DNA, this gray one up here. Now you've got the orange and red. Strand exchange takes place together with RecA. Once that's done, RecA comes off, and now you've got this double-stranded piece, which has a 3' end that your normal polymerase can extend. If you look here, this is the heteroduplex RecA bound. This is your normal heteroduplex over here. You can see they're very different relative to each other. So when RecA is bound, you can't have polymerases. The polymerases are not going to recognize this structure. It's a really bizarre-looking structure. So that has to come off before you can actually get your replication take place. And that seems to be the quality control aspect, is once you have the base pairing that takes place, which is only happening because of the homologous DNA, then you have the RecA, which comes off. You could basically write this slide identically. If you just cross out the RecA here and put rad51, that's the eukaryotic equivalent here. And rad from radiation, because these are radiation sensitive. What does radiation do in terms of DNA damage? So UV irradiation causes thymidine dimers. But what about ionizing radiation? Double-stranded breaks, and that's why radiation medicine is a really good way of doing cancer therapy. In fact, I just met with a radiation oncologist from OHSU on Friday. Daughter's in the same German class as mine. Yeah? Right, so RadA is a single-stranded DNA binding protein, but only binds in the context of this homologous recombination, and the same thing is true of rad51. So it's all a single-stranded binding protein, but it only binds in the context of homologous recombination. The other single-stranded binding proteins bind when? In replication, exactly. Okay, so that's what these guys look like. This is what happens when you have a double-stranded break, again, probably due to ionizing radiation. Again, this is just a different way of looking at it, slightly different colors. Apologies to the red-green colorblind. Some of the males who are here probably. Double-stranded break. Again, you chew in 5' ends, giving you these 3' ends. That then binds to RecA rad51. You have strand invasion here, and what happens is literally you bounce one of those strands off. You move off one of the strands that's not binding to this strand which is invading, together with RecA rad51. The archaeal protein is called RadA, which makes it even more confusing. So you have this strand invasion process, you have extension of this strand, then you displace that other double strand which is there. That's actually very conveniently now going to be homologous to this other 3' end over here, which can bind, extend. And then what you have are these strange-looking DNA intermediates here that we're going to look at in a lot more detail later on, called Holliday Junctions. And that's you've got 2 double strands that are kind of connected to each other in the middle by one strand that goes from one of these double strands to the other double strand, sort of an X structure. We'll look at that in a lot more detail as we move along. These get resolved, i.e. chopped apart and put back together, ending up with fixing your double-stranded break here. Now, this could be, again, due to some kind of DNA damage, but it also happens in meiosis. And that was getting back to your question at the back here, the diploid versus the haploids. So here, in meiotic recombination, when the sister chromatids pair and during S phase, there are specific proteins that are expressed which make double-stranded breaks. And so these are specific proteins, again, made in S phase, that when you've got sister chromatids that are paired with each other, they're going to make double-stranded breaks to get recombination to take place. And so meiotic recombination is actually critical. If you block meiotic recombination, you don't get full progression through S phase and cell division, which is why one of these guys is called SPO11, sporulation. Sporulation happens in yeast when you go through meiosis, because you go from diploid to haploid. So in meiosis, you have to have this recombination that takes place. If you don't have one of these, it's actually an endonuclease stimulation protein, then you don't undergo meiosis. So here, again, just like what you have in double-stranded break repair, once you've made this double-stranded break, you chew in at 5' ends, you've got free 3' ends. RecA, of course, it's not RecA here because these are eukaryotic systems. This is Rad51, strand exchange, DNA synthesis, completely normal. So the only difference that's happening here with meiotic recombination versus double-stranded break repair is you have specific proteins which are endonucleases that will cut in the DNA and make double-stranded breaks. Yeah? Okay, so the question is basically, I'll paraphrase a little bit, but what is Rad51 binding to? When it's not in meiosis. So you'll have double-stranded breaks that'll happen either during replication, which is happening, also in yeast cells. You know, they replicate just as well, so you end up with forks and problems in those forks. So Rad51 will bind to that. Or, again, if you have radiation damage, some kind of ionizing radiation, which will give you double-stranded breaks. And then what you do is the first thing that happens is actually an exonuclease. I should go and grab my model over here again. That chews in at the ends. And then that chews in at 5' ends. So you've got a free 3' end. And this is kind of a giveaway what the problem is here. Binds at the 3' end of your single-stranded piece of DNA. So this piece is missing. Your single-stranded piece, that binds to that piece. That's where Rad51 is going to bind. So Rad51 is going to bind to the single-stranded piece that's made after you have a double-stranded break.
But what's the homologous strand that it then locates if it's not on the homologous sister chromosome?
Okay, so the idea is then there will be something which is homologous. That would either be a sister chromatid in meiosis, but if you're undergoing replication and you've got some kind of problem with replication, then you have another strand which is right there. It's not a sister chromatid, it's just that piece which you've just replicated. And that's what's happening in the bacterial cases, or the haploid genomes, where you have this homologous recombination that's taking place. Yeah?
Are the nicks that are created by these endonucleases are they site-specific?
Okay, I'm not quite sure I follow your question. So the nicks, are they site-specific for meiotic recombination? It's in general.
Provide these breaks in order for recombination to occur, and I'm asking if where those breaks are initiated is really tightly specific and regulated.
Right, specific and regulated. So I think what you mean is when you're making these double-stranded breaks rather than the nicks. Yeah. So these double-stranded breaks seem to be pretty random, and that seems to be important. Again, if you think about meiotic recombination, one of the ideas of meiotic recombination is that that's getting rid of deleterious mutations. Well, you don't know a priori where those deleterious mutations are. And so it had better be pretty random. Now, the problem is, how do you keep it from cutting everywhere, which would be kind of a mess? And the answer is, at least as far as I know, and according to the textbooks, we don't know. So you get a few of them. It seems you have to have a couple of these meiotic recombinations that take place, or the double-stranded breaks and then being repaired, but not too many. And how you know not to have too many is really an interesting question. And we don't, as far as I know, understand quite how that's happening. So this is that process here where we have sister chromatids, duplication and meiosis, and then either crossing over, which is sort of the classic case that you think about in terms of meiotic recombination. You actually can see the chromosomes crossed over relative to each other. or you also have a number of cases what's called gene conversion, and that's where red has been exchanged for blue on either of these. And we'll see how that happens. It actually has to do with how this meiotic recombination process takes place. And I'll give you a, sort of the punchline right now. It's because you're copying from that other copy. And so that's where you're coming from. It's homologous but not identical when you're talking about sister chromatids. Yeah?
So we're talking about meiosis, so gametes. Is this repair happening after that first round of cell division or after the second round?
Oh, so the question is when is this happening in meiosis? Is it happening after the first division or after the second division? I'll let you think about that, because if you think about, it's got to be sister chromatids that are then interacting with each other. Yeah. Okay, so let's look at this again in a little bit more detail. This is how you can also think about how you can get crossing over versus repair, or also what you call patches. So we have our normal meiotic recombination. You end up with these single-stranded ends. Again, this is the 3' end. It's bound to rad 51. Then you have extension with your normal DNA polymerase. This gets to the end here. You have ligation that takes place. Here we have 3' end gets extended. This guy ends up ligating to this guy over here. And now you've got these crossed over structures. You're always going to have two of these Holliday junctions when you have double-stranded break repair, and that double-stranded break repair is also what's happening in meiosis, because you have that endonuclease which has chopped those two strands. So you end up with these two Holliday junctions. Now, the decision here that makes place, so it doesn't actually really seem to be a decision, it seems to be about half and half most of the time, is how you cut and religate these crossed over pieces of DNA. So if you cut them both in the same way here, and so you cut here, this orange will attach to this red, orange attaches to red here, orange attaches to red here, orange attaches to red here, you end up with a chromosome that has both ends that are the same as they started out. So the yellow one up here is still yellow at both ends. The red one at the bottom is red at both ends. If, however, you have one of these Holliday junctions which binds in another way, which actually, as we'll see, these Holliday junctions are completely symmetric. If you have this Holliday junction that instead of binding these two pieces together, binds these two pieces together and these two pieces together, now you end up with one of these crossing over events whereby you end up with now red information at this end and yellow information at this end. And then, of course, because these are reciprocal, this one's going to be red at that end and yellow at this end. So that's the crossing over that takes place. And this is all dependent on exactly how you take those Holliday junctions, cut them, and religate them to each other. And we'll look at this hopefully ad nauseum for the next 5-10 minutes or so. Got to start out with what RecA and rad 51 look like. Again, there were some questions about this before. This is now our DNA, single-stranded DNA binding protein. There's a single-stranded DNA here. These are each of the, I think this one's a RecA structure, but the rad-A structure looks basically identical. Here, if you start out with a single-stranded DNA and a double-stranded DNA, it's just the red for the single strand, the green for the double strand here, incubate them together with RecA, and you can do this in vitro in the absence of anything else. As long as you have ATP around, you end up exchanging the strands. And so you've gone from two greens here to green and red and a single strand in terms of your green. And so this is what happens the very first step. This is that strand invasion process that takes place. After you've had strand invasion, extension by the polymerases, then you end up with these Holliday junctions. Usually people draw Holliday junctions like this as sort of an X'd over form of your DNA, but of course DNA is double-stranded, all wrapped around each other, so it really looks something like this down here in the lower, I guess, right corner for you guys. So this is what these Holliday junctions really look like. Here's another way of looking at that. There are a number of extra proteins that I'm not going to talk about their names of, so I don't expect you to remember them, which are important for binding to these Holliday junctions, these basically, X forms of DNA. And it's a little hard to see here, but if you look at the red strand, this red strand is on one side here, sort of going across that Holliday junction in one way. The light blue strand is going across here. There's a purple strand and a blue strand here. It's a little hard to see. But basically, these double strands here are all made up of different combinations. You've got 4 strands that come in, and then they're exchanging with each other. This is actually looking at a real one of these in the electron microscope. That's what these processes look like. But if you look at this, in fact, it's a little bit easier to look at, I think, this way, thinking about your resolution. So how do you cut the DNA and bind it back together? You really have 2 options here. You can either bind this piece right here to that one over on the other side, or this one over here. And you can also look at it down here in this structure. So you can either, when you need to bind these 2 together, you can cut this guy and bind it to this guy over on the other side, or this guy here on the same side. And they're basically the same place. It's just where you bind and cut these together. And it does seem to be pretty random, sort of half and half, the way that this happens. So as a reminder here, this will give you either no crossover, which is where you have the 2 chromosomes are the same on either side, or crossing over that takes place. And if you like, we can go through this in office hours and look at each of the individual strands, etc. But take-home message here is you can get either crossover or non-crossover, and it depends on how you take those Holliday junctions. If you cut them both in the same way, either here shown horizontally or both vertically, you'll end up with no crossover. On the other hand, if you cut one vertically and the other horizontally, and it doesn't matter which one it is, then you'll have the crossing over that takes place. And this, of course, generates these different chromosomes. So crossing over, again, 2 Holliday junctions have been resolved in different ways. That's pretty obvious how this happens. But then you also have these areas of what's called gene conversion. Now, what's gene conversion? Well, it's gene conversion because this is mostly a red chromosome, but it's got some of this blue chromosomal information in it. And if it turns out that this blue chromosomal information, which is from that sister chromatid, has a different sequence in it, what you're going to end up with are mismatches. And these mismatches are just because it's a differ' sequence on one side than it is on the other side. Now, that's just because of the way that homologous recombination works. Remember, it's the 3' end that you've gotten from one of the sister chromatids that extends. If in that sequence there's some mismatches, you're going to end up with mismatched DNA after you've done your homologous recombination resolution of Holliday junctions. Well, what do mismatches do? They cause structural problems in the DNA. Those are recognized by the normal mismatch repair machinery. It takes out that piece, and it's actually the excision process is a lot like nucleotide excision repair. So it cuts out a pretty big piece and then copies what's left. But unlike the case that you have mismatch repair right after replication, where you know which strand is the old one and which strand is the new one, here they're just as old. They're from sister chromatids. So the mismatch repair machinery doesn't know which strand is the right one to use and which one is the wrong one to use. And so you'll end up with, in about 50% of the cases, you've now taken the information that you had from blue here, and you've now converted it to the information that you had in red. So you'll end up with your non-normal segregation that you'd have normally in meiosis. You'll have two of your parental alleles from one parent, 2 parental alleles from the other parent. But if it turns out there's a region where you had this homologous recombination that took place, then you can end up with 3 copies versus 1 copy. So it's not 2-to-2 segregation. It can be 3-to-1 segregation. And this is that gene, what's called gene conversion process. And those are a lot of these horizontal, vertical lines actually here. The blue areas here in the red chromosome and the red areas here in the blue chromosome. Yeah?
Explain what you just said about instead of having 120, it's 3 to 1. I don't understand.
Yeah, well, so the idea here, so now you've undergone meiosis, right? And so you've got 4 double-stranded pieces, 2 mostly from mom, 2 mostly from dad, but there'll be some regions in them which are partly mom, partly dad. And so you can have gene conversion in one of these, and so you move from being maternal information to paternal information. And in fact, it can happen all the way. All 4 of them could be paternal information, or all 4 could be maternal information. It's just these are pretty random occurrences. Again, it just has to do with how mismatch repair is taking place. And so it doesn't have to be your normal 2 to 2 segregation. It could be 3 to 1 or 4 to 0. And this is how people figured this out in the first place. Actually, we're looking at these things and go, whoa, wait a minute. This makes no sense. We've got these genes that are just not segregating normally. So how could you explain that? And this turns out to be the explanation for some of those. There's other explanations for that. Follow your genetics course to see what's going on there. Okay, so that's it for, the homologous recombination. Just want to talk about one other kind of repair that we haven't talked about yet. And this is basically if things are really, really, really bad and you have to replicate across some kind of problem, replicate, or for that matter, also transcribe. And this is the really quick and dirty process, because what happens is you have these what are called translesion polymerases. And what happens with those is you have normal replication. So here's your normal replication at the top, sliding clamp, DNA polymerase. This will hit some kind of DNA damage. Now it hits this DNA damage, and hopefully that DNA damage gets repaired. But if there's a whole bunch of DNA damage that's in the cell, there may not be enough proteins, enough of the machinery to be repairing all of that DNA damage before cell division is going to take place or something like that. So what happens in some of these cases, again, when you've got a whole bunch of DNA damage, is these so-called translesion polymerases come along. They also associate with the sliding clamp, but they will just add whatever nucleotides next to this DNA damage, just so that you can get past this point of DNA damage to continue your replication. And then hopefully, again, totally over-anthropomorphizing here, the DNA damage repair machinery will come in and take out these DNA damage and hopefully bring it back to what it was in the first place. But it may well be that this translesion polymerase that puts in whatever may end actually putting up the wrong nucleotides there, which when they get copied with DNA repair, particularly with something like nucleotide excision repair, there's going to be the wrong nucleotides which get added. So these are also known as error-prone polymerases, because they will end up giving you errors in your double-stranded DNA. For E. coli, these error-prone or translesion polymerases are activated by RecA. Well, RecA is that single-stranded DNA binding protein that's helping out with double-stranded breaks. If you've got a whole bunch of double-stranded DNA breaks, you're going to have a lot of RecA around, and that then will say, hey, we've got a whole bunch of problems with DNA damage. Let's just get through all of this stuff, finish replication, and then go back and deal with everything else later on. Or very often what happens in eukaryotes is you have a cell cycle block. And in cell biology, talk a lot about control of the cell cycle, where you have particular blocks in the cell cycle. One of the proteins that's very involved in that is called ATM, because when it's mutated, you have a particular disease. And that's one of the ways that you lead to this error-prone polymerase. And we actually know quite a bit about it. We've got a number of crystal structures of these enzymes. Just an example of that here, we've got a thymidine dimer, which has not been repaired. Again, thymidine dimers cause a big change in the structure of the DNA. Your normal DNA polymerase comes along, hits one of these bizarre stretches of template. What do? Okay, we bring in one of these translesion polymerases or error-prone polymerases. It puts in a couple of nucleotides. But if you had these error-prone polymerases that stayed on the DNA, then they'd be putting in whatever nucleotides all the time. So do you want these to be processive or non-processive? Non-processive, exactly. You want them to fall off. And one of the ways you get them to fall off, turns out that these polymerases don't interact with the sliding clamp. So just like all the DNA polymerases, they're not processive unless they have some kind of processivity factor that's associated with them. So that's repair. Do we have any questions, more questions about DNA repair? I'm not going to go over this slide. It's kind of considered as a review. It's a great place to go before the next midterm, which is going to be here before we know it. Okay, so let's talk about transposons, jumping genes. Basically three, I always like to call them flavors, three different kinds of transposons. These are, okay, there are a few exceptions to this, but these are the three main kinds. Either the so-called DNA-only transposons, the retroviral-like retrotransposons, which of course are by far and away the coolest, and then our non-retroviral retrotransposons, which might also be derived from viruses. People argue about that. I give you one guess what side I come down on there. So basically, these guys, DNA-only transposons are just like what you think. They just move via DNA, and we're going to talk about the cut and paste mechanism for how they move around. The retroviral-like retrotransposons, not surprisingly, use reverse transcriptase, and that reverse transcriptase uses an RNA intermediate and looks actually a lot like how retroviruses replicate. So we'll talk a little bit about how retroviruses replicate. These non-retroviral retrotransposons also use RNA intermediates and reverse transcriptase, but they use a slightly different mechanism for this process. And the way that you can tell which one of these you have has to do with their ends. And that's why it's sort of emphasized on here. If you look at these DNA-only transposons, you notice they have these little arrows pointing in opposite directions at either end. Those are what are called inverted repeat sequences. And what that means is if you have a sequence going from 5' to 3' on one strand, then that same sequence going from 5' to 3' on the other strand is an inverted repeat. And so going this way and going that way, that's your inverted repeat sequence. And we can look at those. If I had a whiteboard here, I'd be drawing them up. But for some reason, this room doesn't have whiteboards, and I'm not going to draw with Sharpies on the wall. So on the other hand, if you have direct repeat sequences, which is what you have on these retroviral-like retrotransposons, that's the same sequence on the same strand. So if you've got CATG at one end of your retrotransposon, you've got CATG on the same strand of your retrotransposon at the other end. Again, always going from 5' to 3'. The non-retroviral retrotransposons actually look a lot like messenger RNAs. They've got poly A and, of course, poly T, because this is double-stranded DNA now, tails, and very often the 5' end here is actually not a full-length gene, or for that matter, a full-length transposon. So, okay, why do we care about transposons? Well, that's because 50-ish% of our genome is made up of transposons, particularly these line elements, sign elements we'll talk about a little bit more. The lines are classic non-retroviral retrotransposons. These retroviral-like elements, again, 8 to 10% of our genome, are these things that are clearly very closely related to retroviruses. Interestingly enough, in eukaryotic cells, and particularly in the human genome, there are very few of these DNA-only transposons. On the other hand, in bacteria, there are tons of them. And we know a lot about these DNA-only transposons in bacteria because a lot of them carry antibiotic resistance genes. So in fact, this is the way they were originally found in bacteria, was antibiotic-resistant bacteria that were being picked up in You guessed it, hospitals. So it turns out that the reason they have these antibiotic resistance and how you get antibiotic resistance spread is because of these transposons. Many of them will carry these resistance genes. Like one of the very first ones to be found was this TN10, tetracycline resistance, also ampicillin resistance. Lots of these contain a transposase gene, and that's what the gene is you need, encodes the enzyme to get things to move. They've all got these inverted repeat sequences. That's what these little red boxes are here at the end. So how do these guys move? Again, best understood in bacteria because that's where they were found in the first place. You have a transposon in your genome. Genome is supposed to be represented here by the green stuff on the outside. Here's our transposon in the middle with its little short inverted repeat sequences at either end. This piece in the middle usually encodes a transposase, so it's the gene for the protein which is going to move this gene. It's going to get it to jump. So these guys bind to these inverted repeat sequences. We make a zome because all molecular biologists love zomes. What's zome mean, again, usually for molecular biologists? A complex of stuff. What particular kind of stuff? Proteins and nucleic acids. So you got proteins and nucleic acids together. That's what the molecular biologists love to call zomes. So the transpososome, I hate that terminology. Actually, it's not used that very, very much. But here we've got all of these transposase proteins interacting with the DNA and basically bringing them together, bring both ends together of that transposon. Then these transposase enzymes are endonucleases, so they cut the DNA here, and many of them also will religate that DNA. So now you've got the chromosome piece where you started, and then this transpososome will jump to somewhere else in the genome. And where that somewhere else in the genome is, in 95% of the cases, this transposase will bind to a pretty nonspecific sequence and cut it, but it cuts it not opposite to each other, not base pairs right opposite, just offset by a couple of bases, anywhere from 3 to 6 bases relative to each other. Once it makes that cut, then it links this inverted repeat sequence, which is what the transposase bound to in the first place, to one of those ends, the other end to this end here, and you end up with this transposon, but it has a little gap in it right here. That's what these black things are supposed to represent here, because it was offset at the ends. So it's offset at the ends, you cut, and you end up with a little gap at either end. Those gaps are now next to your transposon, which has jumped in. You've got a 3' OH, you've got a template, plenty of DNA polymerases around in the cell, so they will go in and fill in those ends. But because of that, this sequence that's between where these two cuts were now gets repeated, because it's that extra piece. There's this extra offset piece. That offset piece now is going to get copied at one end by your normal 5' to 3' polymerase, but it's also going to get copied at the other end by 5' to 3' polymerases. And so you end up with a directly repeated sequence, because there's no change in that sequence now, that's outside of your inverted repeat sequences. And this is really important because when you're looking at genome sequences, which we can now do really cheaply, really fast, you end up with lots of repeated sequences that are spread throughout your genome. Well, if you see in that repeated sequence, you have inverted repeats, and then right outside that is a directly repeated sequence that tells you that this is a DNA-only transposon. And so this is a transposon. It's a jumping gene. It's something which is moved into your genome. And so just by looking at the sequence, you can tell that this is what has just happened. You've had one of these cut and paste things that's jumped into the DNA. What does this look like? Here's a crystal structure of one of these transposases attached to DNA ends here. This now is the process that moves, is what jumps will go and bind somewhere else in the genome, moving these transposons along. So now we need to give one lecture of virology in 4 minutes. So this is retrovirus replication. We'll talk much more about it again in virology next term. I think also Monday, Wednesday, Friday from 9 to 10. So just keep doing the same old thing. So retroviruses, classic example here is HIV. We'll ignore all of this really cool stuff that happens here and just say that in a retrovirus replication, you have an RNA which gets released inside the cell. That RNA gets copied first into DNA, and then that RNA is degraded and a DNA is made. And it turns out that the reverse transcriptase here is actually different than the telomerase, because reverse transcriptase in retroviruses is also a DNA-dependent DNA polymerase, not just an RNA-dependent DNA polymerase. So the reverse transcriptase here will make a double-stranded copy of the RNA that came in. This DNA now will get integrated into the host chromosome in a pretty random manner. Again, not dissimilar to these DNA-only transposons that are jumping around. And then this integrated DNA gets transcribed into RNA. That RNA encodes the proteins that then put the virus together, and the virus goes out, and this whole thing goes around again. One of the things that I'm ignoring here, and again we'll talk about a lot more next term, is that this process of going from the RNA to the DNA generates direct repeats at either ends of this DNA copy that goes into the genome. So those are those long direct repeat sequences that you see in the retroviral-like retrotransposons. So unlike the inverted repeats that you have in the DNA transposons, these are the direct repeats that you have at the end of those. What do these enzymes look like? Mostly, in fact, because of HIV, we have a huge amount of really good research on these things. Here's that reverse transcriptase here. Also has an RNase H attached to it. Makes sense to have an RNase H. What do RNase H's do? They degrade RNA when the RNA is in a RNA-DNA hybrid, exactly, which is what you have right here. And that allows that DNA to be made. The actual polymerase, of course, obvious to all of you here because you're all experts on DNA polymerase structures, actually looks really, really similar to all the other DNA polymerase structures we talked about so far. So the actual structure of that enzyme is extremely similar. It's got hand, thumb, fingers, etc. So this is the process, how it's making that. How do these guys get integrated? Integration actually for these retroviral-like retrotransposons and also for retroviruses themselves is extremely similar to what happens with the DNA-only transposons. You have a cut that happens in your target chromosome, a little bit offset at either end. You attach these ends to that. You end up with these gaps. These gaps get filled in, so you end up with short direct repeat sequences on the outside of your viral DNA or the retroviral-like retrotransposon, which is jumped in there. So here we've got direct repeat outside of your sequence, a long direct repeat inside the sequence, and a direct repeat at the other end. So the big difference between these two, if you're just staring at the genome, is just what's on the inside of your transposon. The inside of your transposon, if it's a DNA only, is an inverted repeat. It's a direct repeat if you have one of these retrovirus-like ones. The only one we haven't talked about so far are these non-retroviral-like retrotransposons. Still very open area of research trying to figure out what's going on here. These are normally, again, present in the genome. Always going to have a chicken and egg question. My answer to chicken and egg is always virus. But now we have a copy of this in the genome. It gets made into RNA with a nice poly A tail, normal messenger RNA. Turns out this messenger RNA encodes usually 2 proteins, but we're just going to talk about the reverse transcriptase. This reverse transcriptase binds to the poly-A tail on its own messenger RNA. That then goes and finds a random piece of DNA and cuts it as long as you've got a stretch of T's here, and it can just be 1 or 2, that can now bind to your poly-A tail. The reverse transcriptase activity here can extend and copy its own messenger RNA. People also talk about it a lot as selfish genes, because basically all it's done is it's made a protein which can copy more of itself to make more genes, to make more of itself, etc. That copies the RNA. There's RNase H activity. This then makes a second strand, and you end up with a new copy which is present in the genome. Many cases, what happens is that this particular reverse transcriptase, it doesn't have a nice sliding clamp. It doesn't have a good processivity, so many times it'll fall off of the RNA and then make the second strand of DNA before it gets all the way to the end. That's why many of these genes are truncated at their 5' ends. And so you don't have the full messenger RNA in the genome. So if you look at all of the line and sign elements that are present in the human genome, most of them are truncated. Most of them are actually non-active, which is probably a really good thing that these things aren't jumping around our genome all the time. Although there are a few cases, and we'll talk about them in molecular genetics next term, where actually these do move around. A lot of cases of spontaneous hemophilia actually have to do with these genes once they've jumped around. So there are questions on transposons. Yeah?
The reverse transcriptase discussed in the previous section, does it, I know it cuts it out, but is it also responsible for the ligating back into the
Okay, so the question basically, I think, gets back to this slide, which is, and again, I'm paraphrasing your question. Correct me if I'm wrong here. Does the reverse transcriptase also have the activity of binding, cutting, and religating? The answer is no. In this case, it's an integrase protein. It's actually a separate protein that's involved in doing this. So it's different than the transposase that you have in the DNA only. Transposase binds, cuts, and religates. On the other hand, for these retroviral-like reverse transcriptase and also the reverse transcription that happens excuse me, in these non-retroviral ones, it's a separate protein that's involved in that process. Yeah, so it's the integrase here. Yeah?
Determining whether a repeated sequence is within or without a transposon, is that just experimental of where does the thing cut itself and move?
So the question is, how do you know, basically, when you're looking at sequences, if it's one specific thing or not? One of the things that you'll see is these repeated sequences, the ones you've got hundreds and hundreds and hundreds of them in the genome. Those are all identical to each other. Right next to it is going to be a sequence which is repeated, but that's not identical all over the place. It's just a repeated sequence that's either at end. So you find a repeated sequence, and then you look right outside that, and then you see what's there. Okay, the other, again, maybe sort of emphasis again, thank you for bringing this up, is that if you look at now these non-retroviral retrotransposons, these don't have repeated sequences right next to them. So there's no repetition that happens in that case. Okay, spend the last couple of minutes talking about site-specific recombination because viruses do it, so it's totally cool. But also it turns out to be really important for a lot of some of the newer techniques that people are using in cell biology to knock out genes, use them, etc. And the basic idea here is getting back to the whole idea of inverted repeated sequences or directly repeated sequences. If you have 2 directly repeated sequences on 2 different pieces of DNA, and you have the activity of an enzyme which is extremely similar to those transposases that we just talked about, what it does is it cuts one of these strands, hooks it up to the other one, cuts one of these strands and hooks it up to the other one. This is the process that happens in a particular bacterial virus called Lambda, where it's best understood. But that then cuts and ligates, gives you this directly repeated piece of DNA. with your inserted piece of DNA in between them. The enzyme itself, in this case it's called the lambda integrase. You also have excisionase. You can either have two pieces of DNA here, if you've got directly repeated sequences, or one piece with this thing which is integrated in the middle of it. If, however, you just happen to have inverted repeat sequences relative to each other, so opposite strands, the same enzyme will bind to this piece of DNA, but instead of integrating or excising, it just inverts it, that piece of DNA. So directly repeated DNA, insertion, deletion, inverted repeats, inversion. And if you look at this, just following the DNA around, here we've got X crosses over, goes to A, goes to B, goes to Y. Here, if A and B line up with each other, here A would cross over to B, goes to A and out this side, or you invert, you've got A going to B if you go back in the opposite direction. So direct repeats, inversion, excision, inverted repeats, inversion. We won't talk about lambda because we're going to talk about that next term, so we'll skip over that. Just wanted to talk about how some of these genes are being used for doing genetic engineering or genome engineering. Classic example here that's used very, very often in cell biology is what's called the Cre-Lox system. And this Cre-Lox system, basically what you do is you have a gene that you basically want to knock out, that you put two repeated sequences on either side of. You want to delete this gene. So are these going to be directly repeated or inverted repeats? Direct repeats, exactly. So these direct repeats are going to interact with a protein very similar to the one that's in those DNA-only transposons. And if that gene is around and is expressed, then it will bind to these directly repeated sequences and chop out the gene that you don't want. This is extremely useful if you're trying to get the idea of what a gene is doing in one particular cell type or one particular time during development. What you can do is you end up making a transgenic organism, usually in this case it's going to be a mouse, where you have the gene that you want to get rid of has these two direct repeats on side of it. And then you have another piece of DNA which has this Cre recombinase gene that's only expressed in the right time and place. So when that's expressed, you get the messenger RNA that's made into protein that will bind to this construct and chop this out. This is really important because at other times and in other tissues, you don't make this recombinase gene. And because you're not making this recombinase gene, this gene is made normally. So this is a way you can study genes that would be lethal during development when you actually have something which is happening later on. So jumping genes, we've got our DNA-onlys, we've got our retroviruses or retrovirus-like retrotransposons, the other retrotransposons. We actually kind of skipped over lambda here, but we talked about inverted repeats, direct repeats, and this recombineering is just another way of saying what happens with this expression of Cre recombinase and knocking out specific genes. So with that, I'll see you on Wednesday.